Good afternoon, everybody. I want to thank the organizers of OIS for the invitation to provide an update on ONL Therapeutics. Appreciate the first post-lunch introduction, Rob. Thank you very much. Appreciate it. Want to give you a good update on ONL and where we are. ONL Therapeutics, David Esposito, the CEO. ONL is a privately held biotech company based in Ann Arbor, Michigan, where our technology originated from our founder's lab, Dr. David Zaks, who's a retina specialist at the University of Michigan. Over the last number of years, we've been focused on building an industry-leading platform for neuroprotection. Our lead compound, ONL-1204, across a number of indications, we've seen solid clinical data through a unique mechanism of action of inhibiting the FAST receptor. ONL-1204, with regards to geographic atrophy, we're seeing slowing lesion growth, study eye compared to fellow eye at 3 and 6 month dosing, and slowing consistent lesion growth in those treated eyes. In open-angle glaucoma, we're seeing with ONL-1204 structure and functional improvement in less than 12 months. And we see the real opportunity to bring IOP-independent neuroprotection to the market for glaucoma. And then with regards to rhegmatogenous retinal detachment, our only acute indication, we're going to be reporting top-line results in April, May of next year with, again, 1204 to really look at difference in that acute indication on top of standard of care surgery. The backdrop from all of this, and I think the session on platform, we feel the unique mechanism of action of inhibiting the FAST receptor provides us an opportunity across a number of diseases of the eye. FAST is an upstream activator of cell death and inflammation. ONL-1204, a small peptide delivered via IVT injection, blocks the FAST receptor. By targeting FAST, we prevent that activation of those downstream death pathways. We quiet the inflammatory response. And importantly, in conditions like GA, FAST is upstream of the complement pathways, so we feel it positions ONL-1204 very well to support improved efficacy and durability in patients with geographic atrophy. Importantly, over the last few years, in each of those 3 indications, we've demonstrated target engagement on the FAST receptor with ONL-1204, so we feel with a great deal of conviction, we can move the programs forward across those indications. I want to just highlight some data from both geographic atrophy and also open-angle glaucoma. This outline represents our phase 1b study in GA. It's fully enrolled. It's just about to complete study right now. There are 2 components to that study. The top part, component 1, was a very straightforward open-label dose escalation arm, a single injection of 1204 over 6 months. And then the second component is we're observing patients for 6 months in a natural history phase and then treating with 2 injections of 1204 90 days apart. This is the first data set from that open-label dose escalation component. Again, a single injection of ONL-1204 over 6 months. You can see the 6 patients, study eye, treated eye in blue. Orange is the fellow eye, to show on average across these patients a 42% slowing of lesion growth in treated eyes compared to non-treated eyes over the course of a single injection with 6 months. Again, we think it demonstrates strong efficacy and also important durability in that patient population. This is results from the second component of that study, the first 5 patients who have completed the study, a very similar schematic looking at study eye or treated eyes in blue to untreated eyes, fellow eyes in orange. What you'll see is very consistent across each of the treated patients. ONL-1204 slows lesion growth, study eye compared to fellow eye. This is just in a 6-month time period with 2 injections delivered every 90 days. Again, we think it shows strong efficacy and also the kind of durability that we think is needed in the market. So we continue to progress in our GA program. We're getting ready for a registrational study upcoming in 2024. Also, given the basis of this forum on platform technologies, we wanted to touch on how we think we can change the game in glaucoma based on some of the discussions earlier about looking for neuroprotection beyond what is IOP lowering. Our mechanism of action can support that kind of treatment of an IOP-independent treatment for glaucoma. We start to see now the emergence of a number of different clinical trial designs that could demonstrate the potential of neuroprotection in 18 months to 24 months. We're beginning to build and set the stage for a phase 2 study based on our phase 1 results that I'll be first to declare in this audience here, our first cut from our open-angle glaucoma phase 1 study. This is the outline of the study. It's a 9-month study. We're looking at progressing glaucoma patients, either through a rate of visual field loss with a 12-month look back to make sure they're progressing at least at minus 0.5 decibels a year, or a rate of RNFL thickness loss over a 12-month period, at least 3 readings to show at least minus 1 micron per year loss of retinal nerve fiber layer. We're looking at safety and then also visual field and structural endpoints. As we've begun to look at this data, we've assessed it a number of different ways, looking at a point-wise approach in visual field, looking at cluster visual fields, in addition to retinal nerve fiber layer. So we're seeing retinal nerve fiber layer increases, a number of different ways to look at visual field. What I wanted to highlight is one of those particular analysis with Ted Garway, Heath's structure to function relationship, looking at OCT in 6 quadrants and then mapping those quadrants to visual field. We've developed a relatively simple output to show if you're increasing in retinal nerve fiber layer and you're increasing in sensitivity of visual field at the cluster level, you would be improving up and to the right on this grid. If you were declining in retinal nerve fiber layer and declining in visual field at a cluster level, you'd be down to the left in this grid. This is the first time to show ONL 1204 treated eyes compared to sham to show not just neuroprotection, but neuroenhancement in progressing glaucoma patients. This grid outlines the results of our first 15 patients. ONL 1204 is highlighted in blue, sham is in black. And what you can see is over a very short 9-month time period, 2 injections of ONL 1204, 90 days apart, monitored 6 months post that last dose, seeing retinal nerve fiber consistent increases at a cluster level and improvements in visual field sensitivity at a cluster level for the vast majority of treated clusters and treated eyes in these patients. You can see in the sham patients in those clusters that they continue to decline in retinal nerve fiber layer and continue to decline in visual field sensitivity. This is the first time we've been able to demonstrate, again, not just neuroprotection, but neuroenhancement with ONL 1204. As we look to the future to 2024, we're looking to raise a Series D round of $100 million to run a registrational study in GA. We're ready to be able to put patients in the trial sometime in the middle time of next year. We'll also be reporting top-line results of our first phase 2 study in retinal detachment, about ARVO time period. And through the course of the year and working through our glaucoma data, we'll be finalizing a phase 2 study in glaucoma by the end of the year, and then ultimately moving into what we hope is the early part of 25 with a phase 2 study in open-angle glaucoma. I thank you very much for your time and attention. Look forward to the questions on the panel. Thank you very much.